In animal and fungal cells, cytokinesis is driven by a contractile ring of myosin 2 and actin filaments, which, as well as constricting cells at the division site, may also help coordinate two other activities required for cell division: the delivery of membrane vesicles and remodeling of the extracellular matrix. The proteins involved in these processes are well conserved, but how they work together to achieve cytokinesis is unclear, as Erfei Bi from the University of Pennsylvania explains.
How these components are put together to actually form a machine that drives cytokinesis remains unknown. So we want to look at how the components are wired together using fractal analysis and yeast genetics.
Bi and colleagues, led by graduate student Caitlin Wloka, initially focused on myo-1, the sole myosin 2 heavy chain in budding yeast. Myo-1 begins to accumulate around the bud neck early in the cell cycle, and at every stage, myo-1 fluorescence recovered very little after the entire ring was photobleached, indicating that there's only a limited exchange of myo-1 between the ring and cytosol. To test whether myo-1 is mobile within the ring structure, Wloka et al. Photobleached half of the bud neck at different stages of the cell cycle, and measured the subsequent recovery of myo-1 fluorescence.
Early in the cell cycle, myo-1 molecules remain mobile at the division site. However, around the onset of cytokinesis, the myosin molecules become immobile at the division site.
Myo-1 became immobilized during late anaphase and telophase, at the time that actin filaments assemble into the cytokinetic ring. However, Wloka et al. Found that actin wasn't involved in myo-1's immobilization.
We showed that in two ways. We use nitroncony to depolymerize all the F-actin in the cell, and we showed that by FRAP, myo-1 remains immobile during cytokinesis. The second approach is we abolish actin ring by introducing a mutation in the foreman gene BNI1, and then we monitor the myo-1 dynamics again, and we showed that myo-1 remains immobile in BNI1 mutant.
Myo-1's immobilization was also independent of its motor activity, as the protein's tail domain, which can assemble into a ring and support cytokinesis, still became immobile during anaphase and telophase.
A headless myo-1 behavior during the cell cycle is very similar to the fullness protein.
However, a series of truncation mutants suggested that a C-terminal region of myo-1 is required to immobilize the protein during cytokinesis. These mutants, which remain mobile throughout the cell cycle and show defects in cell division, delete a domain of myo-1 that's thought to be involved in the assembly of bipolar myosin filaments.
So this data suggests that the type of myosin molecules undergo a cell cycle triggered high-order assembly, and that may be critical for its role in cytokinesis.
Wloka et al. Then turned their attention to other components of the cytokinetic machinery to see how their dynamics compared to those of myo-1. Proteins associated with the actin ring, namely tropomyosin and the formin BNI1, were mobile at the bud neck during cytokinesis, as were proteins involved in delivering membrane vesicles to the division site, such as the exocyst subunit XO84. However, proteins involved in forming the primary septum, a region of the budding yeast cell wall that promotes cytokinesis by pushing the cleavage furrow inwards, did become immobilized at the bud neck during cell division.
The proteins involved in septum formation, such as the Caitlin Sedwick 2, IQ-gap, F-bar, or yin-1 protein, are immobile during cytokinesis. Not only that, their immobility depends on myo-1, but not vice versa.
Chitin synthase 2, for example, which helps synthesize the primary septum, was normally immobilized at the bud neck during cytokinesis, but the enzyme remained dynamic in cells expressing the mobile myo-1 mutants that lack the C-terminal assembly domain.
So this data suggests myo-1 may act as a scaffold for the assembly of a cytokinesis machine.
Formation of the budding yeast primary septum is analogous to extracellular matrix remodeling by dividing animal cells, but it remains to be seen if type 2 myosins have a similar function in other organisms.
Type 2 myosin in C. elegans or Drosophila is also immobile during cytokinesis. Whether or not they also act as a scaffold for extracellular matrix remodeling remains to be investigated. In fish yeast, one of the type 2 myosins has been studied and is quite dynamic during cytokinesis. However, our preliminary study indicates that the other isoform of type 2 myosin in fish yeast is immobile. So in a system with more than one type 2 myosin isoform, maybe one is dynamic, that is specialized for contractile activity, while the other is immobile and is perhaps useful for other functions, such as a scaffolding role. So that's a speculation.
So what do Bi and colleagues want to investigate next?
The main question we want to address in the near future is to determine whether type 2 myosin indeed forms a high-order structure, such as bipolar filaments or mini filaments in vitro or in vivo. If so, what the structure looks like. That's the question we're trying to address.
In the meantime, you can learn more about how immobilized myosin 2 plays a scaffolding role during budding yeast cytokinesis in the paper by Wloka et al., published in the February 4th, 2013 issue of the Journal of Cell Biology.